growing the microorganisms. Followed, the production of protein on a large scale by the use of microorganisms is called as single cell protein. Microorganisms ಅನ್ನ ಯೂಸ್ ಮಾಡಿಕೊಂಡು ಬಹಳಷ್ಟು ಪ್ರೋಟೀನ್ಸ್ ಅನ್ನ ನಾವು ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡಬಹುದು. ಆ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡಿ ಸ್ಕೇರ್ಸಿಟಿ ಆದಂತ ಕಂಟ್ರೀಸ್ ನಲ್ಲಿ ಸ್ಕೇರ್ಸಿಟಿ ಆದಂತ ಪೀಪಲ್ಗೆ ಅದನ್ನ ಪ್ರೊವೈಡ್ ಮಾಡಿದ್ವಿ ಅಂತಾರ we can cure the disease. Followed, so the best example for this is called as SCP. It is called as SCP. SCP is single cell protein. Followed, so cyanobacterium, last year you studied about a kingdom. The first kingdom is Monera, that is therein you have studied about bacteria, then archibacteria and cyanobacteria. Usually the cyanobacteria are called as blue green algae. One of the example for this cyanobacteria is spirulina is there. Followed, and this spirulina is a type of that is a cyanobacterium which can be grown, which can produce more amount of protein, and it can be grown on a raw material of lower cost. Raw material ಅದಕ್ಕೆ ಬೆಳೆಸಲಿಕ್ಕೆ ಬಹಳಷ್ಟೇನೋ ಮಟೀರಿಯಲ್ ಬೇಕಾಗೋದಿಲ್ಲ. ಎಂತ ರಾ ಮಟೀರಿಯಲ್ ಬೇಕಾಗ್ತದಂತಂದ್ರ ಪೊಟ್ಯಾಟೊ ಪ್ರೊಸೆಸಿಂಗ್ ಪ್ಲಾಂಟ್ಸ್, ಈ ಅಂಕಲ್ ಚಿಪ್ಸು, ಈ ಕುರ್ಕುರಿ ಅವೆಲ್ಲ ಮಾಡ್ತಾರಲ್ಲ, ಅದು ತೊಳೆದಿದ್ದಂತ ನೀರದಲ್ಲ, ನೀರೊಳಗ್ ಕೂಡ ಬೆಳೆಸಬಹುದು. ಈವನ್ ಆನ್ ಸೀವೇಜ್, ಡಂಗ್, ಈ ಸಗಣಿ ಇರ್ತಲ್ಲ ಅದ್ರ ಮೇಲೆ, ಸೀವೇಜ್ ಗಟರ್ ನೀರ್ ಇರ್ತಲ್ಲ ಅದ್ರ ಮೇಲೆ ಬಳಸಬಹುದು. ಮೊಲಾಸಿಸ್ ಇರ್ತದಲ್ಲ, ಈ ಫರ್ಮೆಂಟೇಶನ್ ಪ್ರೋಸೆಸ್ ನಲ್ಲಿ ಪ್ರೊಡ್ಯೂಸ್ ಆಗಿರ್ತದ, ಅದ್ರ ಮೇಲೆ ಬೆಳೆಸಬಹುದು. ಸ್ಟ್ರಾ, ಒಣಗಿದ ಹುಲ್ಲು ಇರ್ತಲ್ಲ ಅದ್ರ ಮೇಲೆ ಬೆಳೆಸಬಹುದು. ಅನಿಮಲ್ ಮೆನ್ಯೂರ್, ಈಗ ಅನಿಮಲ್ ಫರ್ಟಿಲೈಸರ್ ಮಾಡಿರ್ತಾರಲ್ಲ, ಬಯೋ ಫರ್ಟಿಲೈಸರ್, ಅದ್ರ ಮೇಲೆ ಬೆಳೆಸಬಹುದು. ಟು ಪ್ರೊಡ್ಯೂಸ್ ಲಾರ್ಜ್ ಕ್ವಾಂಟಿಟೀಸ್ ಅಂಡ್ ಕ್ಯಾನ್ ಸರ್ವ್ ಆಸ್ ಅ ಫುಡ್ ರಿಚ್ ಇನ್ ಪ್ರೋಟೀನ್, ಮಿನರಲ್ಸ್, ಫ್ಯಾಟ್ಸ್, carbohydrates and vitamins. So spirulina is a best example. This spirulina is now available in the market in the form of tablets. They are available in the market in the form of tablets. ಆ ಟ್ಯಾಬ್ಲೆಟ್ ಒಂದು ತಗೊಂಡ್ಬಿಟ್ವಿ ಅಂತಂದ್ರ ಬಹಳಷ್ಟು ಹಸಿವಿನೇ ಆಗೋದಿಲ್ಲ. ಯಾಕಂತಂದ್ರೆ ಇಟ್ ಕಂಟೈನ್ಸ್ ರಿಚ್ ಅಮೌಂಟ್ ಆಫ್ ಪ್ರೋಟೀನ್ಸ್. ಈ ಟೈಮ್ ನಲ್ಲಿ ಅದು ತಗೊಳ್ಳೋದು ಬಹಳ ಚಲೋ, ಬಿಕಾಸ್ ಈ COVID-19 ಅದಲ್ಲ, ಕೊರೋನಾ ವೈರಸ್, ಆ ಕೊರೋನಾ ವೈರಸ್ ಫ್ಯಾಟ್ ಕಂಟೆಂಟ್ ಯಾರಿಗೆ ಜಾಸ್ತಿ ಇರ್ತದಲ್ಲ ಅವ್ರಿಗೆ ಇನ್ಫೆಕ್ಷನ್ ಜಲ್ದಿ ಆಗ್ತದ. ಪ್ರೋಟೀನ್ ಕಂಟೆಂಟ್ ಜಾಸ್ತಿ ಇತ್ತು ಅಂತಾರ ಆಗೋದಿಲ್ಲ ಅಂತ. So the spirulina is being preferred in order to satisfy the need of or deficiency of the proteins. Apart from that, a single cow ಅದಲ್ಲ, ಆಕಳ, ಅದರ ವೆಯ್ಟ್ ಎಷ್ಟು ಇರ್ಬೋದು ಅಂತಾರ 250 kg ಕೌ ಇದ್ದಂತದ್ದು ಎಷ್ಟು ಪ್ರೋಟೀನ್ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡ್ಲಿಕ್ಕೆ ಸಾಧ್ಯದ ಅಂತಾರ, ಇಟ್ ಕ್ಯಾನ್ ಪ್ರೊಡ್ಯೂಸ್ 200 grams ಆಫ್ ಪ್ರೋಟೀನ್ಸ್ ಪರ್ ಡೇ. ನೋಡ್ರಿ, ಕೌ ಬಾಡಿನಲ್ಲಿ ಎಷ್ಟು ಪ್ರೋಟೀನ್ಸ್ ಪ್ರೊಡ್ಯೂಸ್ ಆಗ್ತಾ ಇರ್ತದ. 250 kg ಕೌ ಅದ, ಅದರಲ್ಲಿ ಎಷ್ಟು ಪ್ರೋಟೀನ್ ಪ್ರೊಡ್ಯೂಸ್ ಆಗ್ತಾ ಇರ್ತದ, ಬರೀ 200 grams, ಪಾವ್ ಕಿಲೋನು ಅಲ್ಲಿಲ್ಲ, ಅದಕ್ಕಿಂತ ಕಡಿಮೆನೆ ಗ್ರಾಮ್ಸ್ ಆಫ್ ಪ್ರೋಟೀನ್ ಈಸ್ ಪ್ರೊಡ್ಯೂಸ್ ಪರ್ ಡೇ. ಫಾಲೋಡ್ the 250 grams of 250 grams ಪಾವ್ ಕಿಲೋ microorganisms. ಯಾವ microorganism? The microorganism named Methylophilus methylotrophus. ಮೆಥಲೋಟ್ರಾಫಸ್ ಅಂತ ಹೇಳಿ ಮೈಕ್ರೋ ಆರ್ಗಾನಿಸಮ್ ಅದಲ್ಲ. ಎಷ್ಟು ತಗೊಂಡಿರ್ತೀವಿ? ಬರೀ 250 grams ಆಫ್ ದಿಸ್ ಮೈಕ್ರೋ ಆರ್ಗಾನಿಸಮ್ ಪ್ರೊಡ್ಯೂಸಸ್ ಅಬೌಟ್ 25 tons of proteins. It produces about 25 tons of proteins. 25 ton ಅಂತಂದ್ರೆ 25 quintal proteins can be produced. And the another example for this is mushroom. ಮಶ್ರೂಮ್ ನಲ್ಲೂ ಕೂಡ ಏನಿರ್ತದ ಅಂತಂದ್ರ, ಮಶ್ರೂಮ್ ಅಂತಂದ್ರೆ ಈ ನಾಯಿ ಕಡೆ ಲಾಸ್ಟ್ ಇಯರ್ ಅಗ್ಯಾರಿಕಸ್ ಎಡಿಬಲ್ ಮಶ್ರೂಮ್. ಫಾಲೋಡ್, ಇಟ್ ಕ್ಯಾನ್ ಬಿ ಪ್ರೊಡ್ಯೂಸ್, ಇಟ್ ಕ್ಯಾನ್ ಬಿ ಗ್ರೋನ್ ಆನ್ ದಿ ಸ್ಟೋನ್. That is, raw material is of less expensive raw material, and it contains a rich amount of proteins also. So by growing these different organisms on the raw materials, we can satisfy the need of a protein, or we can satisfy the deficiency of proteins. Followed, and this is called as a single cell protein. Single cell protein yakantivantandra, that is, single celled organisms are mainly involved in the production of protein. So it is called as single cell protein. Followed. So single cell protein is the method of production of large scale proteins by the use of microorganisms. By the use of microorganisms. And best example for this is spirulina. Apart from that, the another example for the another example is Methallophilus methallotrophus, which can produce about, that is only 250 grams of these microorganisms can produce about 25 tons of proteins. So what are these proteins? What are proteins? Last year you studied this one in your biomolecules chapter. Proteins. Anybody? What are proteins? Butila? These proteins are the organic components which are made up of carbon, hydrogen, oxygen, nitrogen, sulfur, phosphorus, etc. And amino acids are called as building blocks of proteins. Followed. These proteins, that is group of proteins, they form the amino acid. They form the amino acid. So amino acids are called as building blocks of proteins. So next one is about tissue culture. So what is this tissue culture? Tissue culture is the method of growing a plant cell or the tissue or an organ, plant organ, in an artificial nutrient medium under laboratory condition to produce a new plant. Followed. I have brought a plant today. This is a plant. The part of the plant, the part of the plant, either the part of the stem having the cell, the part of the root having the cell, the part of the stem having the cell, or the part of the leaf having the cell, it is grown in a nutrient medium. It is kept in a nutrient medium for few days of time, which will grow into a new plant. Followed. That nutrient medium is artificially prepared. So growing of new plants by using the part of the plant under laboratory conditions in the nutrient medium is called as a tissue culture. This process is called as tissue culture. ಈ ಪಾರ್ಟ್ ಅನ್ನ ನಾವು ನ್ಯೂಟ್ರಿಯೆಂಟ್ ಮೀಡಿಯಂ ಹಾಕಿ ಇಂತಹದೇ ಗಿಡನ್ನ ಬೆಳೆಸಬಹುದು. ಮಣ್ಣೊಳಗೆ ಹಾಕೆಲ್ಲ ನ್ಯೂಟ್ರಿಯೆಂಟ್ ಮೀಡಿಯಂ ಒಳಗೆ ಹಾಕಿ ಇದನ್ನ ಬೆಳೆಸಬಹುದು. ಆ ಪ್ರೋಸೆಸ್ಗೆ ಏನಂತ ಕರೀತಾರ? ಇಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಆಸ್ tissue culture. That is the ability of the plant cell. ಈ ಪ್ಲಾಂಟ್ ಸೆಲ್ ಎಂತ ಎಬಿಲಿಟಿ ಇರ್ತದ, ದಿ ಎಬಿಲಿಟಿ ಆಫ್ ದಿ ಎಂತ ಎಬಿಲಿಟಿ ಇರ್ತದ ಅಂತಂದ್ರೆ ಗ್ರೋ ಇನ್ ಟು ಅ ನ್ಯೂ ಪ್ಲಾಂಟ್. ಎಲ್ಲಿ ಸೆಲ್ ರೂಟ್ ಒಳಗಿನ ಸೆಲ್ ಇರ್ಬೋದು, ಸ್ಟೆಮ್ ಒಳಗಿನ ಸೆಲ್ ಇರ್ಬೋದು, ಲೀಫ್ ಒಳಗಿನ ಸೆಲ್ ಇರ್ಬೋದು, that cell has a capacity to grow into a new plant. Followed. So that is the ability of a plant to multiply and regenerate into new plant is called as totipotency. It is called as totipotency. Regeneration ಅಂತ ಹೇಳಿ animals ನಲ್ಲಿ ಹೇಳ್ತೀವಲ್ಲ, ಹಂಗ ಇಲ್ಲ plants ಗೆ ಇದಕ್ಕೆ ಏನಂತ ಕರಿತೀವಿ? We call it as a totipotency. The capacity of this cell to grow into a new plant. The capacity of part of the plant to grow into a new plant is called as a totipotency. Followed. Whereas in case of animals, it is called as a regeneration. And this regeneration capacity is best seen in case of poriferans. Poriferans. A poriferan powder powder to now regenerate ಮಾಡಿದ್ವಿ ಅಂತಾರ, it will grow into a new plant. It will grow into a new organism. Even planaria is there. Followed. But we are supposed to study about the plants here, and the capacity of the plant cell to grow into a new entire plant is called as that is totipotency. Later you can ask the question. Okay, the property of this totipotency led to the scientists to develop a desired form from the explant. So the part of the plant, think for a while, I have taken the leaf, part of the leaf, the part of the plant which is used in the tissue culture. Either it may be from the leaf, or it may be from the root, or it may be from the stem. The part of the plant that is used in the tissue culture, this part is called as explant. It is called as explant. What is explant? The part of the plant, the piece of the plant, followed, which is used in tissue culture, is called as explant. And this explant, any part of the plant may contain a cell, or it may contain group of cell called as tissue, or it may contain organ also, which is that is, it is kept, this explant is kept in a test tube containing nutrient medium. So nutrient medium ಒಳಗೆ ಏನೇನ್ ತಗೊಂಡಿರ್ತೀವಿ? Nutrient medium contains water, organic components, inorganic components, and growth promoters, hormones. The growth promoters like auxin, IAA, indole acetic acid, then gibberellin, GA1, GA2, GA3, ಮತ್ತೆ cytokinin, 6 perfural aminopurine. ಇವು ಎಲ್ಲ ಆರ್ಗ್ಯಾನಿಕ್ ಕಾಂಪೋನೆಂಟ್, ಇನ್ಆರ್ಗ್ಯಾನಿಕ್ ಕಾಂಪೋನೆಂಟ್, ಪ್ಲಾಂಟ್ ಹಾರ್ಮೋನ್ಸ್ ಎಲ್ಲ ಹಾಕಿ ಒಂದು ನ್ಯೂಟ್ರಿಯೆಂಟ್ ಮೀಡಿಯಂ ಅನ್ನ ತಯಾರು ಮಾಡಿರ್ತಾರೆ. ಆ ನ್ಯೂಟ್ರಿಯೆಂಟ್ ಮೀಡಿಯಂ ನಲ್ಲಿ ಏನ್ ಹಾಕಬೇಕು ಅಂತಾರ? ವಿ ಹ್ಯಾವ್ ಟು ಕೀಪ್ ದಿಸ್ explant. Followed, what is explant? This explant is part of the plant which is used in tissue culture, is called as explant. So when this explant is placed into the nutrient medium, what happens? The cells that are present in an explant, after about 2 to 3 days, or after about 1 to 2 days, what happens? The cells start to undergo division. The cells start to divide. Followed, and these cells which have been divided, they result in the formation of undifferentiated mass of cells. The cells divide and redivide and produces undifferentiated mass of, that is undifferentiated mass of cells. And these undifferentiated mass of cells produced by this explant is called as callus. It is called as callus. ಗೊತ್ತಾಗ್ಲಿಕತ್ತದ? ನ್ಯೂಟ್ರಿಯೆಂಟ್ ಮೀಡಿಯಂ ಒಳಗೆ ಹಾಕಿದ್ವಲ್ಲ, ನ್ಯೂಟ್ರಿಯೆಂಟ್ ಮೀಡಿಯಂ ಒಳಗೆ ಹಾಕಿದ ತಕ್ಷಣ ಸೆಲ್ ಏನಾಗ್ತದ ಅಂತಂದ್ರ, ಇನ್ ದಿ ಪ್ರೆಸೆನ್ಸ್ ಆಫ್ ನ್ಯೂಟ್ರಿಯೆಂಟ್ ಮೀಡಿಯಂ ಅಂಡರ್ ಲ್ಯಾಬೊರೇಟರಿ ಕಂಡೀಷನ್ಸ್, what happens? The cell divides and produces undifferentiated mass of cells. That is, it divides and divides and produces mass of cells, and this mass of cells so produced by a single cell or group of cell is called as callus. It is called as callus. The cells of the explant start dividing in the medium and gives undifferentiated mass of tissues called as callus. The callus later gives rise to what it produces. It gives rise to embryoids called as somatic embryoids. That is, it produces what it produces. Later, it produces the descending part and the ascending part. The descending part is called as a radicle, and the ascending part is called as plumule. The radicle develops into root system, and the plumule develops into shoot system. Followed, and the callus later gives rise to embryoids called as somatic embryoids. The embryoids generates into a new plant. New small plantlets are being produced in the test tube. A small plantlets produce ಆಗ್ತಾವಲ್ಲ, that is these small plantlets so produced, they are being transferred. ಎದಕ್ಕೆ ಟ್ರಾನ್ಸ್ಫರ್ ಮಾಡ್ತಾರಂತಾರ? They are transferred in a plastic bag containing, that is a red soil. ಅದ್ರಲ್ಲಿ ಏನ್ ಮಾಡ್ತಾರಂತಾರ? ಸಾಯಿಲಿಗೆ ಅಡ್ಜಸ್ಟ್ಮೆಂಟ್ ಆಗ್ಬೇಕಂತ ಹೇಳಿ ಮೊದ್ಲು ಏನ್ ಮಾಡ್ತಾರಂತಂದ್ರ, ಅದಕ್ಕ ಪ್ಲಾಸ್ಟಿಕ್ ಬ್ಯಾಗ್ ಒಳಗೆ ಅವ್ರು ಟ್ರಾನ್ಸ್ಫರ್ ಮಾಡ್ತಾರೆ. ಆ ಪ್ಲಾಸ್ಟಿಕ್ ಬ್ಯಾಗ್ ಒಳಗೆ ಏನಾಗ್ತದೆ ಅಂತಂದ್ರೆ, ಅದು ರಿಚ್ ಗ್ರೋತ್ ಆಯ್ತು ಅಂತಂದ್ರ, ಲೇಟರ್ ದೇ ಆರ್ ಬಿಂಗ್ ಟ್ರಾನ್ಸ್ಫರ್ ಟು ದಿ ಗಾರ್ಡನ್ ಸಾಯಿಲ್. ಗಾರ್ಡನ್ ಒಳಗೆ ಲ್ಯಾಂಡ್ ಒಳಗೆ ಹಾಕ್ತಾರ, ಯಾಕಂದ್ರೆ ಅಲ್ಲಿ ಹಾರ್ಡ್ನೆಸ್ ಬರ್ಬೇಕಂತ. And the plant can be grown, the entire plant can be grown by a technique called as tissue culture, or by a process called as a tissue culture. Followed, so what is the use of tissue culture? ನಾವು ಇಷ್ಟೊಂದು ಪ್ಲಾಂಟ್ಸ್ ಬೆಳೆಸ್ತಾ ಇರ್ತೀವಿ, ಈ ಟಿಶ್ಯೂ ಕಲ್ಚರ್ ಯಾಕೆ ಫಾಲೋ ಅಪ್ ಮಾಡಬೇಕು? ಈ ಟಿಶ್ಯೂ ಕಲ್ಚರ್ ಯಾಕೆ ಫಾಲೋ ಅಪ್ ಮಾಡಬೇಕು ಅಂತಂದ್ರ, ಇನ್ ಆರ್ಡರ್ ಟು ಪ್ರೊಡ್ಯೂಸ್ ಲಾರ್ಜ್ ನಂಬರ್ ಆಫ್ ಪ್ಲಾಂಟ್ಸ್ ಆಫ್ ಎಕಾನಾಮಿಕ್ ಇಂಪಾರ್ಟೆನ್ಸ್. ಒಂದು ಮೆಡಿಸಿನಲ್ ಪ್ಲಾಂಟ್ ಇರ್ತದ, ಆ ಮೆಡಿಸಿನಲ್ ಪ್ಲಾಂಟ್ ಒಂದೇ ಇತ್ತು ಅಂತಂದ್ರೆ ಅದನ್ನ ಹೆಂಗ್ ಪ್ರಪೋಗೇಟ್ ಮಾಡಬಹುದು ಅಂತಂದ್ರ, ಇಟ್ ಕ್ಯಾನ್ ಬಿ ಪ್ರಪೋಗೇಟೆಡ್ ಇನ್ ಟು ಥೌಸಂಡ್ಸ್ ಅಂಡ್ ಮಿಲಿಯನ್ಸ್ ಬೈ ದಿ ಪ್ರೋಸೆಸ್ ಆಫ್ by the process of tissue culture. And such a plant, that is this process is called as micropropagation. Production of thousands of plants from a single plant, economically important plant, followed, is called as, by the technique of tissue culture, is called as micropropagation. It is called as micropropagation. Plants that are grown by this micropropagation process, micropropagated plants ಇರ್ತಾವಲ್ಲ, these micropropagated plants are genetically identical. Genetically as well as morphologically identical species are being produced during sexual reproduction, and they are called as clones. So also here, the plants which are produced, the plantlets which are produced by the technique of tissue culture called as micropropagation, these micropropagated plants are genetically as well as morphologically, they appear to be same, and they are no longer called as clone, but they are called as soma clones. They are called as soma clones, SOMACLONE, yes. Because why they are called as soma clones? Why not they are called as only the clones? That is in order to avoid the confusion. And moreover, these are produced by somatic cells. The root, stem, and the leaves, they contain somatic cells, body cells are there. So such clones are called as soma clone. So this is about tissue culture. What is the application of tissue culture? Where we have to apply this tissue culture? What is the use of tissue culture? So as I told you earlier, this tissue culture helps in production of thousands of plants from a that is from a single economically important plant, commercially important plant. ಒಂದು ಪ್ಲಾಂಟ್ ಇರ್ತದ, ಆ ಪ್ಲಾಂಟ್ ನಲ್ಲಿ ಬಂಗಾರದ್ದು ದಟ್ ಈಸ್ ಫ್ರೂಟ್ಸ್ ಫಾರ್ಮೇಶನ್ ಆಗ್ತಾವ ಅಂತ ತಿಳ್ಕೊಳ್ರಿ, ಪ್ರೊಡ್ಯೂಸ್ ಆಗೋದಿಲ್ಲ. ಆ ಪ್ಲಾಂಟ್ ನಾವು ಬೆಳಿಬೇಕು ಅಂತಂದ್ರೆ ಹೆಂಗ್ ಬೆಳಿಬಹುದು ಅಂತಂದ್ರ, ಇಟ್ ಕ್ಯಾನ್ ಬಿ ಗ್ರೋನ್ ಬೈ ಮೈಕ್ರೋ ಪ್ರೊಪಗೇಷನ್. ಒಂದು ಮೆಡಿಸಿನಲಿ ಇಂಪಾರ್ಟೆಂಟ್ ಪಾಯಿಂಟ್ ಇರ್ತದ, ಆ ಮೆಡಿಸಿನಲಿ ಇಂಪಾರ್ಟೆಂಟ್ ಪ್ಲಾಂಟ್ ಅದನ್ನ ಕಷಾಯ ಮಾಡಿ ಕುಡಿದ್ವಿ ಅಂತಾರ ಕೋವಿಡ್ ಹೋಗ್ಬಿಡ್ತದ ಅಂತ ಹೇಳಿದ್ರೆ ಎಲ್ಲಾರು ಬೆಳೆಸ್ಲಿಕ್ಕೆ ಶುರು ಮಾಡ್ತಾರಲ್ಲ. ಸೊ ಅದನ್ನ ಹೆಂಗ್ ಬೆಳೆಸ್ಬೋದು ಅಂತಂದ್ರ, ಇಟ್ ಈಸ್ ಬೈ ಟಿಶ್ಯೂ ಕಲ್ಚರ್. Followed. So production of large number of plants from small tissues or single cell is called as micropropagation. And micropropagated plants are genetically same or genetically identical, called as soma clones. Followed. And production of disease resistant plants. Production of, we can produce a disease resistant plant by culturing the apical meristem. Apical meristem. Apical meristem is present at the tip region of root and shoot. See, entire plant is a disease now. Entire plant is infected by bacteria and virus. And this is economically important plant. ಈ economically important plant ಅಲ್ಲಿ ಎಲ್ಲಾ parts infect ಆಗ್ಬಿಟ್ಟಾವ. ಸೊ ಇನ್ಫೆಕ್ಟ್ ಆದಂತ ಪಾರ್ಟ್ಸ್ ಅನ್ನ ಬಿಟ್ಟು ಇನ್ಫೆಕ್ಟ್ ಆಗ್ಲಾರ್ದಂತ ಪ್ಲಾಂಟ್ ಅನ್ನ ಬೆಳಿಬೇಕು ಅಂತಂದ್ರೆ ಯಾವ ಪಾರ್ಟ್ ಅನ್ನ ಬೆಳಿಬೇಕು ಅಂತಾರ, ದಿ ಎಪಿಕಲ್ ಮೆರಿಸ್ಟಮ್. Because you know that apical meristem which is present at the tip region consists of meristematic cell, and that apical region always grows. ಆ ಗ್ರೋತ್ ಆದಾಗ ಏನ್ ಮಾಡೋದು ಅಂತಾರ, ಅದೇನ್ ಇನ್ಫೆಕ್ಷನ್ ಇನ್ನ ಆಗಿರೋದಿಲ್ಲ, ಅದನ್ನ ಎದಕ್ ಯೂಸ್ ಮಾಡ್ಕೊಳ್ಳೋದು ಅಂತಾರ, ನಮ್ ಟಿಶ್ಯೂ ಕಲ್ಚರ್ ಯೂಸ್ ಮಾಡ್ಕೊಳ್ಳೋದು. Followed. So disease free plants can be produced by that is culturing the meristem, that is apical meristem, especially to obtain the virus free plants or disease free plants. Then production of haploid plants. See, almost all the plants are multicellular, and each and every cell is having 2n number of chromosomes deployed in nature. So haploid plants can be produced. Hang haploid plants and produce ಮಾಡಬಹುದು ಅಂತಾರ by culturing the pollen grains. Pollen grains are haploid in nature. Pollen grains which are spherical in shape, produced inside the anther, they can be cultured in the nutrient medium to produce a new plant. This pollen grain is haploid in nature. It multiplies to produce mass of cells, and all these mass of cells are also haploid in nature. And those mass of cells differentiate to produce plumule and radicle, and this plumule grows into shoot system and the radicle grows into root system. Entire plant so produce, either it may be the root, or it may be the stem, or it may be leaf. All these parts, they contain these cells having n number of chromosomes. Such plants are called as haploid plants. What is the use of producing these haploid plants? Haploid plants plays very important role in plant breeding. Plant breeding. Method of producing varieties of plants. Varieties of plants is called as plant breeding. So this can be produced. Pollen culture results in the production of the haploid plants. Production of somatic hybrids through protoplast culture. Protoplast culture. Protoplast can be cultured to produce the somatic plants. So what do you mean by this protoplast culture? Wait, I'll first of all I'll tell you one more point. Later I'll come to this somatic hybrids. That is production of secondary metabolites like drugs. You have studied about narcotic drugs, which are of three types. The first one are called as opiates. The second one are called as that is cannabinoids, and the third one is called as coke. These opiates, which act as the stimulants, that is sedatives, I think. That is these are extracted from a plant named Papaver somniferum, poppy plant. Cannabinoids that is are extracted from cannabis sativa, whereas coca is extracted from erythroxylum coca. So these are called as secondary metabolites. Such plants can be produced on a large scale by the technique of tissue culture. Followed. So the secondary metabolites like drugs, alkaloids, etc., can be produced by this callus culture. Hmm? The next one is, as I told you earlier, about this production of somatic hybrids. What are this somatic hybrid? Somatic cell hybridization. It is the method of fusing the protoplasm of two somatic cells of desirable plants to obtain a hybrid protoplasm. ಎರಡು ಪ್ಲಾಂಟ್ಸ್ ತಗೊಳ್ಳೋದು, ಎರಡು ಪ್ಲಾಂಟ್ಸ್ ತಗೊಂಡು ಅದ್ರಾಗಿನ್ ಬೇರೆ ದಟ್ ಈಸ್ ಬಿಲಾಂಗಿಂಗ್ ಟು ಡಿಫ್ರೆಂಟ್ ಸ್ಪೀಸೀಸ್, ನಮ್ಗೆ ಬೇಕಾದಂತ ಪ್ಲಾಂಟ್ಸ್. ಒಂದು ಪೊಟ್ಯಾಟೊ ಪ್ಲಾಂಟ್ ತಗೊಂಡಿರುವಂತ ಕೊಡ್ರಿ, ಒಂದು ಟೊಮ್ಯಾಟೊ ಪ್ಲಾಂಟ್ ತಗೊಂಡಿರಿ. ದಿ cells are extracted from potato plant and the cells are extracted from the tomato plant. The cell wall, as you know that these plant cells are covered by cell wall, which is extremely hard and rigid, as it is made up of cellulose. So this cellulose is being digested by using the enzymes. Other enzymes use ಮಾಡಿ ಆ cellulose ಅನ್ನ ಎಲ್ಲ digest ಮಾಡ್ಬೋದು. ಆ ಸೆಲ್ಯುಲೋಸ್ ಡೈಜೆಸ್ಟ್ ಮಾಡಿದ್ವಿ ಅಂತಾರ ಎರಡು ಸೈಟೋಪ್ಲಾಸಮ್, ಎರಡು ಸೆಲ್ ಒಳಗಿಂದ ಇದು ಒಂದ್ ಸೆಲ್, ಇದು ಒಂದ್ ಸೆಲ್, ಈ ಸೆಲ್ದ ಸೆಲ್ ವಾಲ್ ಡೈಜೆಸ್ಟ್ ಮಾಡಿದ್ವಿ ಅಂತಂದ್ರ ಇದ್ರಾಗಿ ಲಿಕ್ವಿಡ್ ಕಾಂಪೋನೆಂಟ್ ಕಾಲ್ಡ್ ಆಸ್ ಪ್ರೋಟೋಪ್ಲಾಸಮ್ ಎರಡು ಕೂಡ್ಬಿಡ್ತಾರೆ. ಆಯ್ತಾ? ಈ ಕೂಡ್ದು ಅಂತಾರ ದಿಸ್ ಈಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಕಂಬೈನ್ಡ್ ಪ್ರೋಟೋಪ್ಲಾಸಮ್, ಮಿಕ್ಸ್ಡ್ ಪ್ರೋಟೋಪ್ಲಾಸಮ್. ಆ ಮಿಕ್ಸ್ಡ್ ಪ್ರೋಟೋಪ್ಲಾಸಮ್ ಒಂದು ಸೆಲ್ ವಾಲ್ ಫಾರ್ಮೇಶನ್ ಮಾಡಿ ಅದನ್ನ ಕಲ್ಚರ್ ಮಾಡಿದ್ವಿ ಅಂತ. ಅದು ಕಲ್ಚರ್ ಮಾಡಿದಾಗ ಏನಾಗ್ತದೆ ಅಂತಾರ a complete plant is being produced. Remember that it is diploid in nature. A complete plant is being produced. This plant will be having the characteristic of potato as well as characteristic of tomato. ಈ ಪ್ಲಾಂಟ್ ಏನ್ ಮಾಡ್ತದ, it produces potato inside the soil and tomato on the upper surface of the soil. This is called as pomato. This plant is called as pomato, which has a capacity of producing both potato as well as tomato at a time. And such type of culturing is called as somatic cell hybridization. It is called as somatic cell hybridization. Protoplasts of two desirable plants with desirable characters are fused to form the hybrid protoplast either by using electric field or by polyethylene glycer. And such offsprings produced by this method are called as somatic hybrids, and the process is called as somatic hybridization. So this is the applications of tissue culture. These are all the applications of tissue culture and end of this chapter. Followed. So you are having notebook along with you? Notebook along with you? Okay. Okay. Right heading as single cell protein. Single cell protein. SCP, single cell protein. Single cell protein. Production of production of production of protein biomass. Protein biomass. Protein biomass in a large scale. In a large scale. In a large scale. By using the microorganisms. The microorganisms. Microorganisms. And growing them in a low cost, LOW, low cost raw material is called as single cell protein. Single cell protein. Single cell proteins. For example, spirulina, SPIRULINA, spirulina can be grown on wastewater from potato processing plants. Potato processing plants. Comma, molasses, 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 comma, straw, and even sewage water, SEWAGE, sewage water to produce large quantities of proteins. Large quantities of proteins which can serve as SERVE, serve as the food. Change the line. 250 grams of microorganisms like Methylophilus, METHYLO, PHILUS, Methylophilus, Methylotrophus, METHYLO, TOPHUS, TROPHUS, Methylotrophus, produces 25 tons of proteins. 25 tons of proteins. 25 tons of proteins. Next, write heading as tissue culture. Tissue culture. Tissue culture. It is the method of growing a plant cell or tissues 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 or tissues in the artificial nutrient medium ARTIFICIAL artificial nutrient medium under laboratory conditions laboratory conditions under laboratory conditions change the line. The part of the plant used in tissue culture is called as explant. EXPLANT is called as explant. Underline it. Explant. Change the line. The cells of the explant divides in the medium in the nutrient medium nutrient medium and produces undifferentiated mass of cells called as callus. CALLUS called as callus. CALLUS callus. Next line. The ability of the plant cell to generate a whole plant, a whole whole plant is called as totipotency. 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 Next, write heading is application of tissue culture. Application of tissue culture. Application of tissue culture. Application of tissue culture. First one, it helps to produce thousands of plants, thousands of plants from small tissues, or by using single cell, and this method is called as micropropagation. 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 Underline ಮಾಡಿ. Micropropagation. Second point, micropropagated plants, micropropagated plants, PROPAGATED propagated plants are genetically identical, GENETICALLY, genetically identical, IDENTICAL, called as soma clones. SOMACLONES, soma clones. Underline madri. Soma clones, as they are produced by the somatic cells. Somatic cells. SOMATIC, somatic cells. Third one, disease-free varieties, can be produced by culturing the apical meristem, which produces virus-free plants. Next, haploid plants. Next point, haploid plants can be produced by culturing by culturing the pollen grains, pollen grains. Next, production of secondary metabolites, metabolites, metabolites like drugs, alkaloids, alkaloids, through callus culture, CALLUS, callus culture. Write it as somatic hybrids. Somatic hybrids. SOMATIC. Somatic hybrids. HYBRIDS. Method of. It is the method of fusing the protoplast, PROTOPLAST, protoplast of two TWO somatic cells of desirable plants, DESIRABLE, desirable plants, to obtain a hybrid protoplast. Such protoplast can be grown into a new plant, and such hybrid plants are called as somatic hybrids, HYBRIDS, and the process as somatic hybridization, HYBRIDIZATION, somatic hybridization. For example, production of plants like pomato, pomato from potato and potato and tomato, tomato. Unmute your mobile. This is all about the strategies for enhancement in food production. So in our next class, let us study about this microbes in human welfare. Before that, I'll send you questions on this chapter, and you have to note down the answers for that. I think many of the students are not writing the answers. You are just this is a single way. It has become one way. I'm simply they'll be studying the class and you will be hearing it. So students, work hard and write the answers for that. And if you are having any difficulties in it, please call me. And there are some CET questions for you students. That is emasculation. Emasculation of flowers is carried out through a removal of stigma, sepals and petals, anthers, entire organism.
Answer.
It is the removal of anthers. Then next question is, in tissue culture, single germinating pollen grain will form a dash plant. In tissue culture, single germinating pollen grain will form a dash plant. Which type of plant is produced? Which type of plant is produced by culturing the pollen grain? By culturing the pollen grain, which type of plant is being produced? Is anybody? Haploid plant is being produced by culturing the pollen grain, right? The next one is fever line breeds refers to homozygosity and independent assortment, homozygosity only, heterozygosity, and linkage. Fever line plants, fever line capital T ತರ fever line ಅಂತ ಹೇಳ್ತೀವಲ್ಲ, it is produced by homozygosity and independent assortment. So next one is the process in which mature differentiated cells resort to that is reverse to meristematic activity to form callus is called as. The process in which mature differentiated cells reverse to meristematic activity to form the callus is called as cytodifferentiation, redifferentiation, dedifferentiation, differentiation. The permanent cells which have lost the capacity to divide again regains the capacity of division whenever there is a need. That is process is called as differentiation. Is called as differentiation. The next one is, in crop improvement, virus-free clones can be obtained through. In crop improvement, virus-free clones can be obtained through embryo culture, grafting, shoot apex culture, hybridization. It can be obtained through shoot apex culture, that is apical meristem. So next one is artificial hybridization is the transfer of pollen grains to the stigmatic region from a flower with a desired trait, the same flower, any flower, none of the other. That is flower with desired trait. Then Sonalika and Kalyansona. Sonalika and Kalyansona are high yielding varieties of. These are the high yielding varieties of sugarcane, rice, wheat, maize. Sonalika and Kalyansona,
Wheat.
and these are the varieties of wheat. So these are some of the sample questions on this enhancement in food production. And I'll send you all the notes I have dictated all the notes, and I'll send you the questions. You have to answer for that. And let us meet again on Saturday at 8 o'clock. Okay? Any doubts regarding this chapter?
No, ma'am.
No doubts at all. Okay. Okay. Shall I leave now then?